My name is Wang Tao. I'll introduce the paper submitted to iMeta. The title is, Vaginal microbiota are associated with in vitro fertilization during female infertility.
The main causes of female infertility are ovulation problems, polycystic ovary syndrome, and so on. The demand for safer and more effective assisted reproductive technologies increases with the prevalence of infertility. On the other hand, the vaginal microbial community has attracted people's attention, which mainly refers to the group of microorganisms living in the female vagina, which plays an important role in maintaining vaginal health and reproductive health. Research shows that the vaginal microbiome may predict the outcome of in vitro fertilization. Thus, it is important to consider whether IVF outcomes could be influenced by the microbial composition of the reproductive tract among women with different types of infertility.
Here, we analyzed the microbiome of 1,411 people, 1,255, received embryo transfer, and revealed that moderately rich lactic acid bacteria were the most beneficial to pregnancy, exceeding the higher abundance seen in 1 mora and 3 mora. Metagenome analysis of 71 samples shows that non-pregnant women are detected with more antibiotic resistance genes, and proteobacteria and firmicutes are the main hosts. Result 1. Phenotype and vaginal microbial composition. The biochemical and clinical factors of 1,411 females diagnosed with infertility for no pregnancy. The age averaged 31.66 and body mass index, BMI, averaged 22.33, were analyzed. Age and levels of luteinizing hormone, prolactin, and anti-Mullerian hormones were negatively correlated, but positively correlated with follicle-stimulating hormone after multiple testing corrections. At species level, we detected the following species belonging to the Lactobacillus genus: L. iners, L. crispatus, L. jensenii, and L. hominis. We found a significantly widespread presence of Lactobacillus, which was all negatively correlated with the abundance of other 14 genera. Respectively, Gardnerella and Prevotella were significantly positively correlated with 13 and 11 genera respectively. Result 2. Community types of vaginal microbiota in infertility women. To further understand the variation in microbiota communities present in the vagina, we examined community state types, CST, across all samples using the Valencia, a nearest centroid-based classier. This analysis revealed V major groups and 9 subgroups of microbial communities. The taxonomic composition at the species level of samples, designated to each CST, typically aligned with that of the related reference centroid. As anticipated, variations in Shannon diversity were noted among the sub-CSTs. The co-occurring vaginal bacterial networks showed several differences between the 5 community types with samples of more than 180. Result 3. Association of the vaginal microbiota with biochemical and clinical measurements. Estradiol, E2, was found to be significantly associated with the largest number of microorganisms. In women of reproductive age, the relative abundances of lactobacillus were found to be positively correlated with age, suggesting an increase in the colonization of these bacteria in the vagina as females age. Lactobacillus was a biomarker for age. G. Vaginales was a biomarker for weight. Result 4. Differences in the vaginal microbiomes of women suffering various types of infertility. For all 1,411 samples, we recorded whether the patients were diagnosed with any symptoms. Among the 1,231 women with infertility, most of them, 783, 63.61%, were diagnosed with an inflammation condition, namely chronic salpingitis, endometritis, and pelvic inflammatory disease. Next, LEFS analysis was conducted between the control and each infertility group. Only 12 and 7 infertility-associated bacterial toxa were identified in females with low AMH and intrauterine adhesion, respectively. The distribution of different community types in each infertility group was also explored. Type 3A samples showed a high-level distribution of scarred uterus, N equals 81. The highest proportion of Type 4B samples were from women who were diagnosed with salpingectomy. Result 5. Association of vaginal microbiota composition with IVF outcomes. Research shows that only age, AMH, and LH had a significant effect on the pregnancy rate. Type I and Type II contain the largest number of women. Lactobacillus jensenii is a biomarker for this CST, showed the highest pregnancy rate. When we checked the difference between pregnant and non-pregnant women in the CST, we found that the lactobacillus was a biomarker for the pregnant women. Result 6. Schematic infertility vaginal microbiome from metagenome perspective. We performed shotgun metagenomic sequencing of the genomic DNA of 71 vaginal samples. After metagenome assemble and taxonomy annotation, our results show that the majority of the contigs were assigned to Lactobacillacea, Bifidobacteriacea, and Prevotacea. At the genus level, the vaginal flora was also dominated by Lactobacillus, followed by Gardnerella and Prevotella. On the other hand, Gardnerella showed a significantly higher abundance in non-pregnant women. At the species level, Lactobacillus jensenii exhibited a relatively higher abundance in pregnant women. That Lactobacillus jensenii was a marker for CST3B with the highest pregnant rate. Compared to the pregnant women, the non-pregnant women contained more annotated genes and antimicrobial resistance genes, ARGs, than pregnant women. Our final summary is as follows. Pregnancy rates vary among women with different vaginal microbiome communities. Too high abundances of both Lactobacillus crispatus and Lactobacillus iners have negative effects on in vitro fertilization (IVF) outcomes. A moderate abundance, around 80%, of Lactobacillus is more beneficial for pregnancy. These findings may aid the development of strategies for predicting vaginal microbiota to control IVF outcomes.